Bio Penguins. Sorry for that delay, but we should be live, and we are doing Unit 4 review over cell communication and cell cycle. If you've been in my previous lives, you know that I've been sick, and I'm, guess what, still sick. So if I happen to cough, I'm so sorry, and if I have to take a sip of water again, I'm sorry. But I am doing my best. We, of course, are here, ready to rock and roll, and do some cell communication and cell cycle. So, as you all know, you are AP Bio Penguins. You are here. You are ready. You are going to be dressed for success. No matter what happens on that exam on May 4th, you are ready. You are going to do your best. You're going to leave it all on the test, and you're going to rock it like a penguin. So, in case you don't know, there are tons of resources available for you. So I post daily on my Instagram story. I started on February 2nd, and I'm currently on 5.2, I think is what we did on Friday. So you can tune in. If you missed it, I've saved the PowerPoint for each of those on my website, as well as it's saved in like the highlight section. And if y'all have questions as we're doing this live, just so you can type them in the chat. I see y'all have been talking back and forth as I had my little delay there. So y'all are welcome to ask questions there, and you can help each other in the chat. I have a 505-page review guide that I posted on my website. It takes you through each of the standards within the CED, and it allows you some I can statements, like if you can do these things, then you should be able to meet the requirements within the CED or the curriculum for AP Bio. There's also topic questions in there, as well as I have multiple choice and free response questions aligned to each unit, broken all up in there for you. At the end, I've gotten all the numeric responses. I have all the graphing questions all together. And then there's, tips and tricks throughout the whole thing. I have FRQ Fridays, so I've done every FRQ from 2013 to 2024. And I've made a video for each of those. So if you're doing an FRQ and like, hey, how should this be scored? Or what could I kind of, help out with, or what would be my, okay, I know nothing, where can I get points if I don't know anything? And so sorry, y'all. So those FRQ Fridays can kind of be a little supporting piece for you. If you're not ready to sit down and write an FRQ, you could play one of those videos. You can pause it, think of what you think the answer would be, and then you could play the video and see if your answer is there. I've also got over 120 Wayground games that you can play to kind of help you to, do quick check on your content, as well as their review videos and PowerPoints on my Weebly. And again, that's apbiopenguins.weebly.com. Honestly, if you Googled AP Biology, AP Bio Penguins, you will probably find the Weebly account. So 10 to 15% of the exam is on Unit 4. And again, Unit 4 is cell communication and the cell cycle. So we're going to go through a little bit of cell communication. We're going to do signal transduction pathway and the feedback process. And then there's some cell cycle as well as the regulation of it. Mitosis and meiosis are split up into two different units. So today we focus on mitosis, and then Unit 5 is when we'll do the meiosis as well as the heredity that people are currently talking about in the chat. The person in the question about Unit 3 is photosynthesis and cell respiration. We did that one 2 weeks ago on March 1st. And that video is also saved if you need that. So let's get into this. So cell communication. So one thing is that we can go through direct contact. And so cells have these things called glycolipids and glycoproteins that are on their cell membrane. And so the receptors can come directly in contact with one of those glycoproteins or glycolipids that are on their membrane. So through physical connections, they can allow signals to be passed directly between the two, okay? We also have a little bit of synaptic signaling, chemical signaling. This can be our short distances as well as our long distances. So short distances means that we're sending those materials, those signaling molecules, we're going to later call them ligands, into the surrounding air, into the extracellular fluid, and then they're going to bind to a cell that's nearby. Now, the cells that are going to respond to them are going to be the ones that have the appropriate receptor. So our receptors are proteins that are going to be on the membrane if it's a protein hormone or protein signal, or like some type of polar signal. If it's nonpolar, that receptor is on the inside. But so what's going to happen is that if it has the right receptor. So the receptor is, of course, protein, as we said, is going to have an active site for that ligand. And so if it has the right size, shape, charge, then that ligand will be able to bind to it. And we also can have long distance. So like hormones, they can get into the bloodstream and they can go all over your body. And so that'll be more of a long distance kind of signaling. Okay. So when we look at our short distance, we could talk about autocrine. Autocrine means that the signal is going to be released from the cell and it's going to bind to the same cell. So auto makes us think of self or me. And so with autocrine, it's going to bind to itself. It's a little bit faster for it to go through signal transduction pathway than it is for it to like diffuse through the cytoplasm, because diffusion rates are kind of slow. We also have paracrine. Paracrine is, of course, going to go to a nearby cell. This is looking at gap junctions. And so if you remember, there's a gap junction that occurs between our animal cells. It's just like a little passageway between two neighboring cells. It's also called a plasmodesmata if you're looking at plant cells. And then endocrine has to do with that we're doing long distance, it's going into the bloodstream and traveling all around. And as I said, it has to do with that there's going to be some type of receptor on that cell that has the right active site for that ligand. And so someone's asking, if you haven't started studying for the exam, is it too late? Nope, not too late. We still have 50 days for that AP exam. And if you go to my website, I have an 8-week and a 4-week review kind of schedule. Combine the two and make yourself a one in the middle, because we're approximately 6 weeks away from the exam. Is it 7? I don't know how many weeks it is. So signal transduction. There are 3 steps that we have for our signal transduction. The first step is reception. So with reception, your ligand, also known as your signaling molecule, is going to bind to the receptor. And once it binds, as we know multiple times, if you bind anything to a protein, it always changes shape. And so when that receptor binds, let me see if I can do this. Okay. So if this was like my signaling molecule, right? When it binds to the receptor, okay, you're not going to have just wibbling around, right? It's going to change shape. That's called the induced fit, right? Whenever you bind to a protein, always changes shape. And so it changing shape is then going to be the first step that we see for transduction, okay? So an example that we have would be the G protein coupled receptor. There was a G protein coupled receptor in the I'm going to go 22, it may be 2023, FRQ question. It was on cholera. And so if you're looking for like, hey, I need an example of this, which actually I think we're going to do that example later on in this session. And so we'll do, so the first step is that conformational shape change. It changes shape. And that then, of course, starts the whole process. Okay. Now we have different types of ligands. There could be a steroid hormone, or there could be a protein hormone. So steroids, we talked about those in Unit 1. And the things you can think about steroids, you're like, oh, wow, for a few drinks, cool. But they're non-polar. That was the big thing we have to remember about those steroid hormones. So because of the fact that they're non-polar, they're going to be able to move directly across the plasma membrane. And so they move across the plasma membrane, and their receptors are going to be intracellular. Intracellular meaning it's going to be in the cytosol or inside of the nucleus, traditionally going to be inside that cytosol. Okay, so some examples we might see is testosterone or estrogen. Now, on the exam, they're probably going to have to tell you this is a steroid hormone. They're not just going to tell you a name and assume you know which ones are steroids and which ones are proteins. Okay, so I'm just giving you examples, but they would have to give you some type of information that would make you know that, or they may tell you that the receptors on the inside are an intracellular receptor, and you have to know, oh, well, that would be a steroid hormone. Okay, so you might have to make a little bit of connections here. For a protein hormone, this is going to be released by exocytosis. Proteins are made in the rough ER that then goes to the Golgi, that then goes to the plasma membrane. We've talked about exocytosis. And so that's how we will release these protein hormones. And so their receptor is going to have to be extracellular. Why is it extracellular? Why is it sitting on that plasma membrane? And the reason being that it can't get across the membrane. So since the membrane is nonpolar and the protein is polar, it cannot move across that membrane. And so the receptor must be on the outside. And so an example of this would be insulin. And so here you can see kind of the overall process for signal transduction. We have a ligand, and it's going to bind right here to a receptor. Okay, so that's what we're looking at right here with our reception. Okay, this part right here, this is all part of transduction, which we're going to look at in just a second. There'll be some type of signaling that goes through and binds then to this enzyme called adenylyl cyclase. And based on this image, and I'm going just based on the image, you can see the ATP is converted into cyclic AMP, and oh look, now we have protein kinase A gets activated, and there must be some type of response that comes from that. This whole process is looking at the full signal transduction pathway. So as we said, transduction, this is this part in the middle, okay? It's just going to be like a domino, okay? There are all these relay molecules that are in there. You can have secondary messengers. That should definitely say secondary messengers. I'm so sorry about that. So we have all these things in the middle that's just going to bring it from reception to response. That's all that's happening in transduction, is just to relay the signals. It helps to amplify it. So like you can have one relay molecule that then, activates multiple relays, that then activates multiple relays. So we can amplify that signal. So I know with like epinephrine, we end up getting like 10 to the 8th molecules of like glucose released from the cell. Like, so we kind of see that there is that amplification that takes place. And so we can go through a phosphorylation cascade. Based on the name, you can tell, oh, phosphorylation cascade. There must be something getting phosphorylated. Okay, and so the enzyme we see there is a kinase. So protein kinases are going to phosphorylate our relay molecules. Traditionally, they're going to be proteins. So remember, enzymes tell you what they do. So a protein, this is going to be something that's going to phosphorylate protein, so protein kinase. We also have secondary signals, right, or secondary messengers. So again, this hormone should say messenger. I'm so sorry. We have calcium as well as cyclic AMP. So here in this example, you see that cyclic AMP. So adenylyl cyclase is converting this ATP into cyclic AMP, and then the cyclic AMP continues on doing something. So these are usually going to be like very small molecules that are able to move throughout the cytosol or the cytoplasm. This calcium, we see that sometimes with our muscle cells. So within muscle contraction, you have your calcium stored in your sarcoplasmic reticulum, which is a type of smooth ER. And so when the signal gets to the smooth ER, it then causes the calcium to be released. The calcium then binds to something else inside the cell. You don't need to know about it, but the troponin and the tropomyosin complex, which then allows for the binding, which then gives you your muscle contraction. And so, as I said, they're just little molecules that are moved throughout to then allow for the next part of the signaling, which is the response. And so our last step would be our response. Different things can happen. It's all based on the diagram that they give you. So different cells are going to have different relay molecules. So even if you have the same signaling molecule, you could have a different response to it. And so whatever they give you on the exam, they will have to give you some type of diagram. They'll have to give you some type of explanation, something that lets you be able to go from part A to part B. So the different options, the cell can grow. We can allow it to secrete something. So gene expression, it can be a transcription factor and turn on or turn off gene expression, or turn on or turn off making of a certain thing. We also can alter the function or alter the phenotype, the physical characteristics of it, or it can lead to apoptosis. Apoptosis is programmed cell death, and so it can cause the cell to basically digest from the inside out, and so it will get rid of that cell. And for those of you that are freaked out about that, your hands were originally a big blob of cells, and then you went through apoptosis, and that's what allows you to have the spaces in between your fingers. Also, you had a tail while you were in development, and then apoptosis, and we were able to remove that tail, or like it kind of, disintegrated. So as a reminder, there are 3 steps that we have for signal transduction. We have reception, where we're going to receive the signal. There were 2 different types, steroid and protein hormones. And so it might be important to know the differences between those to be able to know, oh, this is this type of hormone, so this is where the receptor will be. We also have transduction. This was our next part. This is just the cascade. It's the kind of the domino effect, if you may, of how it's going from one place to the next, like to get us from the reception to the response. And then here's our different options for our response of what can happen as a result of that signal binding to that cell. So keep on rolling. It doesn't look like there's been a lot of questions. Just the only question I saw was endocrine long distance signaling. Yes, endocrine signaling is long distance signaling. That would be with your hormones in your bloodstream. Okay. There we go. And the other questions we'll probably get back to later because they're just about the exam. And so we have 2 different types of feedback. We have negative feedback and we have positive feedback. So let's first look at negative feedback. So the big part about negative feedback is it's trying to maintain homeostasis. It's trying to get us back to normal, okay? So it's going to reduce that initial stimulus. The goal is to kind of, if our temperature gets too high, it brings us back down to normal. If our temperature gets too low, it brings us back up to normal. How can storage be a response? I don't see storage as a response. Storage, I'm not really sure what you mean by storage. I'm assuming if you're talking about storing of like glucose, yeah. So with the insulin molecule, the insulin can bind to a cell that can then cause the cell of the liver to take up the glucose, and then it can convert it from glucose into glycogen. So that could be a response. I'm giving you the responses that are directly out of the CED. And so I don't believe that they're going to ask you storage on the AP exam because that wasn't one of the ones that was given in the CED. And so there would have to be some information that they gave you that would make you think that storage was a response. Do you have to know the mechanics about cyclic AMP? No, you do not need to know about the mechanics of the cyclic AMP. Every time I've seen cyclic AMP on the exam, there's always been some type of diagram or some type of description. So back to our negative feedback. So if you're like, for example, if your blood glucose level gets too low, your body releases insulin, which will then bring your glucose level backwards. If your blood glucose level gets too low, you release glucagon, which brings you back up to normal. Or if your blood glucose level gets too high, you release insulin, which then brings you back down. And so that's all part of that negative feedback that homeostasis is maintaining. Okay. So if the system gets disrupted, the negative feedback's main job, again, is to return it back to that set point. And this can either be the molecular, the cellular, or the organismal level. Okay, and this is straight out of the CED. And so what we see with these feedback loops is that whatever the final product is going to come back and it's going to inhibit. So on the exam, you might see a diagram like this. And if you see a straight line like this, that shows you that something is being inhibited. So whatever this product is coming back and it's inhibiting, and it's going to decrease the process of that signaling, okay? And so here we can see a specific example about threonine, and you can see we have this, pathway. And so if we have enough of this isoleucine, and I don't need to make any more isoleucine, then we're going to see negative feedback. The isoleucine is going to come back and it's going to bind to enzyme 1 to inhibit it. It's going to act as a, some type of inhibitor. I'm not really sure if it's going to, well, it says it binds to allosteric site. So this will be considered a non-competitive inhibitor because it's going to bind to a different site, which will be the allosteric site. On that enzyme 1. Does that mean I stop making isoleucine? No, we'll still make it, but in very small quantities. And so due to the fact that the process still takes place on its own. And so if they were to ask on this question, that you would either say there was no production of A, which then of course causes you to not make isoleucine, or you can see there would be less production of A, which then of course decreases the production of isoleucine. There's those two options depending on how you look at that enzyme functionality. So I hope there's no questions on negative feedback. I haven't seen any come through. So positive feedback, this is going to amplify your product, okay? So most of the pathways we see are usually, and again, if you hear random noise in the background, those are my kids, just go with it. So positive feedback, most of our stuff is negative, but we do also have positive feedback that takes place. This is going to be an amplification. It's going to build up. It's going to get us to some type of product. It's going to go in the same direction as your the product, okay? And so usually what's going to happen is it's moving away from that set point, okay? And so in this case, our product is going to come back and it's going to amplify. It's going to cause that stimulus to keep happening. It's going to keep the building up of this process, okay? Now, we have an interesting example for this one. So with this one, this is looking at childbirth. And so what's happening is that the baby's head is pushing on the cervix, which then causes the release of this oxytocin. Oxytocin is then going to cause uterine contractions. And so as the uterus contracts, it pushes the baby's head even harder onto the cervix, which then causes even more oxytocin to be released. And so we're going to see that there's this buildup of oxytocin, which increases the contractions. And so then we're going to see the release of the baby. So do we need to know at least one positive, one negative feedback? Give an example. Positive feedback we just did, and negative feedback. So what's going to happen on the exam is probably going to either tell you about a pathway, and it's going to ask you, is this positive or is this negative feedback? Or they're going to ask you, why would this be negative feedback or why would this be positive feedback? You're going to have to use some given example. So I don't really think you need to know an example off the top of your head to give them the example, but you have to be able to apply this concept. And so by me showing you examples, it makes it a little bit easier for you to be able to apply in a future way. Plus you can also see what it means by negative and positive feedback. And so again, negative feedback, it brings us back to normal, okay? And so as we see that product, it's going to come back and inhibit this pathway versus positive feedback is going to keep building up. And y'all, if you take chemistry, don't forget to go check out Chem with Corinne. She is phenomenal. I love her so much. Anyway, so But again, negative feedback is going to bring us back to normal. It's going to regulate, bring back. Okay. Yes, and positive is going to move us away from the homeostasis. It's going to build up to get to some type of product. We also see the oxytocin with the milk release. So in a lactating mother, the oxytocin causes the milk to be released, and as the baby continues sucking, we're going to keep releasing that milk to keep feeding the baby. Um, but so if the baby stops sucking, logically you're not going to keep releasing the milk. And so that's kind of where we see by it's building up until the signal isn't there anymore. So I hope that was good. Okay, so on to cell cycle. So now the cell cycle has multiple different parts. Okay, so everywhere from here all the way over to the other G, this is all considered interphase. So we have interphase, and then we have the mitotic phase. Okay, and so it's important that you know what's happening in each of these steps of our cell cycle. And so the cell cycle is just the life of the cell, basically the life cycle of one cell. So in G1, the cell is going to be metabolically active. It's just chilling. It's doing its thing. It's a normal cell. Yay. Sorry, y'all. So this one is going to duplicate its organelles as well as the cytosolic components. Okay. In the S phase, this is when we're going to go through synthesis, and we're going to synthesize that DNA. That's when DNA replication takes place. We'll talk about DNA replication in Unit 6. So at this exact moment, you don't really know how it happens. Just know that it does happening. And so we'll make a copy of the DNA at this point. And then in our second gap, this is when we're going to synthesize the proteins. We produce a large amount of ATP for the process of this division that's about to take place, as well as the centrosomes are going to replicate. The centrosomes are going to your microtubule organizing centers, and since microtubules are needed for the mitotic phase, this is just when we're going to, of course, make 2 copies of those, so we'll be able to then later on move them to the opposite poles for the process that will take place in mitosis. And then, of course, the mitosis is going to be, or the M phase is our nuclear division. And so mitosis is the nuclear division, while cytokinesis, which we'll talk about in a second, is the cytoplasmic division. And so, that's kind of important to know. Okay. Now, if a cell is no longer dividing, okay, then it's going to go over to this thing called G0 or G naught, okay? And that's the non-dividing state. Is just the cell being a cell, chilling, doing its normal thing, okay? Some of our cells are like that. So your liver is in G0, your muscle cells are in G0, and then your nerve cells are in G0. And I don't remember exactly what age that's going to start, but they're in G0. The reason why you can bodybuild is because your muscles actually get bigger, your muscle cells are getting bigger, not that you're going through mitosis and making more of them. So someone says there's 2 checkpoints. Yeah, there's going to be a checkpoint in our G1. There's a checkpoint in our G2, and then there's a checkpoint in the M phase. We deal with the checkpoints when we get into the next section, which is on the regulation of it. So there are going to be, we'll talk about the checkpoints in the next part. I kind of worked through the CED and kind of was truncating based on the different parts of the CED for this. Okay, so check to make sure mitosis, yeah. And so it means by duplicating organelles, for example, the mitochondria. The mitochondria, we're going to need to have more mitochondria for the 2 different cells. We're going to have to make more of all of our organelles so that we'll be able to have enough for 2 cells. Because again, this one cell is going to become 2 cells. So we need to make more of those organelles to be able for each cell to have the appropriate number of organelles. That's what they mean by duplicating organelles. So mitosis, it has a role in growth. So this is how you're able to grow. You have, of course, more cells, they grow, and that's what it let you be this infant child to now this adult, or actually you're not really adults, you're more like teenagers, but yeah. So into these teenagers that we now have, okay? Tissue repair. So I always give the example because we have this huge set of stairs outside of my school that somebody fell down the stairs and you skinned your knee, right? Are you going to forever have a gaping hole in your knee? No, you're not going to have forever a gaping hole in your knee. Your cells are going to replicate. They'll go through mitosis so that we can kind of seal up the bleedingness that you've got going on, those platelets, all that fun stuff. And then we'll also, fix the skin so we'll be able to have a nice, beautiful little knee again. And then also asexual reproduction. So looking at our organisms that are I'm going to go through asexual reproduction. So think about grass goes through asexual, bananas are asexual. So thinking about how those different organisms are going to be able to make another copy of themselves is by mitosis. Okay. So same thing as synthesis phase, but the organelles, yes. So it's just the exact same thing. We're making more copies of those organelles. Okay, so prophase. So I have a stupid dance that I've always done with this, and like, you're gonna laugh at me in a second when I do this, but that's okay because that's why we're all here is to learn, right? Yay, cool. Okay, so we have prophase, metaphase, anaphase, and then telophase. Okay, so that is the parts of mitosis, and then after mitosis we go through cytokinesis. Yeah, you weren't expecting that. I know that. It's okay. It's cool. Anyways, so it goes prophase, metaphase, anaphase, telophase, cytokinesis. Okay, and so that's the different phases that we see with the mitotic phase. Over the years, students have like modified. They're like G1, S, G2, and then they go prophase, metaphase, anaphase, telophase, cytokinesis. Okay, you got the idea. Anyway, so in prophase, what's happening is that our sister chromatids are going to condense, okay? So all of this DNA, right, we've replicated in the S phase. Now there's a complete copy of all that DNA. And so we've got, the original DNA plus its duplicate. Well, that's what we call sister chromatids. Sister chromatids are going to be the chromosome plus its duplicated chromosome. And they're going to pair up together at this little place in the middle of them called their centromere, okay? And so they're going to condense down and make themselves into those little chromosome shapes because we don't really want the free-floating DNA that's uncondensed. And they're going to kind of find one another. So I always say like prophase preparing, because we're going to prepare to divide by our sister chromatids condensing. Our mitotic spindle is going to form, and those centrosomes that I mentioned that are holding our microtubules are going to move to opposite poles. So you can see right here, these are our centrosomes. They've moved to opposite sides. You can also see that our nuclear envelope is disintegrating. It's going away. I need to be able to pull those chromosomes to opposite sides. And you can kind of see the microtubules are kind of starting to come out, but they haven't really connected anything. Okay. So then we go into metaphase. Metaphase is going to be when our spindle fibers, those microtubules, they're going to attach to our centromeres, right? They're going to attach to the kinetochore. Oh, I said that wrong. Sorry. They're going to attach to the kinetochore that's at the centromere. And there's going to be this tug of war. And that tug of war gets them to be in the middle. So that's why we move our hands to this phase. So like this was prophase, we prepared. Then we went into metaphase. So this is now lining up in the middle of the cell. And for middle, you can do whatever you want. But I've made my hands perfectly straight like this because it's showing that they're in a straight line in the middle of the cell. Now, some of you learned something called prometaphase. Prometaphase is just looking at that transitional process between moving from prophase into metaphase. It was that tug of war that we're seeing as it's moving down to the middle. Condensing just means that it's going to basically ravel up. And so chromatin is made up of DNA, and then there are these proteins called histones. And so the DNA is going to wrap around the histones. I kind of think of it like a fork and spaghetti. It like wraps around it so that it can condense, because you don't want to try to separate a whole plate of spaghetti. But if you wrap each of your spaghetti noodles around an individual fork, you can easily split those across your plate and evenly have equal amounts of your spaghetti or your chromosomes on each side. Do sister chromatids also refer to the two halves of the chromosomes in the meiosis? I'm not really sure what you mean because in meiosis, we're looking at homologous chromosomes, so it would actually be 4. So it's sister chromatids, and then these would be non-sister chromatids together, but it's all homologous. But we'll look at meiosis in our next unit, okay, like when we get to the next one, okay? So what we see here is, again, metaphase. We have our microtubules are attached to the kinetochores. We're right up in the middle of the cell. So then anaphase, right? So now we're at this point, right? Anaphase, they're going to move apart, okay? So the reason I move my hand like this is because on the side of it, you've got that microtubule pulling from the middle. So it kind of makes this little like C shape, if you may, from them, okay? Because at this point, we've already separated the two. There was an enzyme called separase that came through, and it separated your sister chromatids from each other, and that allowed that when the microtubules pulled from the opposite sides, these sisters would then pull apart from each other, okay? And so I have one set of chromosomes going to one pole and the other set of chromosomes going to the other pole. Okay, and so it pulled your pair of sister chromatids, it separates them towards the opposite poles. Sister chromatids will have identical genes and alleles. The homologous chromosomes are actually different. Yeah, so homologous chromosomes would be like the maternal and the paternal of chromosome 1. That would be your homologous pairs. But since we're in mitosis, we're just looking at the sister chromatids. And so they're all lined up individually and they're not as a pair. Okay. And then telophase is, or some people call it telophase. As long as you spell it right, no one's going to know how you pronounce it. Anyway, so telophase is going to be when we form our two new nuclei. So here you kind of have separated. We form the new nuclear envelope around it. The mitotic spindle is going to break apart. And then, of course, After this is when we go into cytokinesis, which is when we divide that cytoplasm, okay? And so this will be different based on the different types of cells. So with an animal cell, we are going to have a cleavage furrow. So you're going to have an actin ring, if you remember, microtubules, which were part of your cytoskeleton. They're going to did I say microfilaments? Anyways, the actin filaments, these are microfilaments. They're part of the cytoskeleton. They're going to basically take a ring and they're going to make themselves smaller and smaller and smaller until finally, like, you have two different cells. The cell plate, oh my God, that should be our plant cells. I'm sorry, y'all. The cell plate is in our plant cells. And so what's going to happen is it's going to form this little, like, they're going to drop off pieces of like this primary cell wall. And that will then separate the two cytoplasm apart from each other. Okay, so questions. Do you need to know what kinetochores are and memorize their function? Kinetochore is not directly stated in the CED, so they may mention them in a question, but they cannot expect you to know kinetochore. Based on my teaching of my students, I always make sure I explain it to them, but I don't know if they're going to ask you specifically about kinetochores. This is where the cleavage furrow forms an animal plant. Yes. Okay, so I think we've said everything on that, so we're good there. So somebody brought up earlier about the regulation, about knowing the different checkpoints. And so the cell cycle, there's a lot of things that can go wrong. And we want to make sure that everything is good so that we can appropriately separate our cells. We do not want there to be a process where now I don't have enough chromosomes, or my chromosomes weren't correctly separated apart, or they're not actually done being replicated. So we want to make sure that we are able to make identical cells at the end of mitosis. So the regulation are just looking at these different checkpoints, these different controls that take place to basically as a stop, like hold up, wait a minute, put a little bio in it, to make sure that everything's good. So our G1 checkpoint is going to determine whether we can complete the cell cycle. Do we have a signaling molecule called a growth factor that says, hey, I need you to grow? So for example, when you fell down the stairs and you skinned your knee, your platelets are going to release something called platelet-derived growth factor. That's then going to stimulate for you to make more platelets to then seal up that area and, make it so that you don't have a big hole in your knee, right? So do we have a signal that says, hey, I need to make more copies of this cell? Do I have that signal? Do I have enough reserves? Is there enough resources that I have that I need to be able to, of course, divide? And then I'm going to check for my DNA damage, okay? If there's a problem in your DNA, do you want to make a copy of it and then replicate that cell? No, you do not want to do that, okay? So if it doesn't pass the G1 checkpoint, if we don't get the appropriate signals to do that, then we go into that G0 or that G0, which is our non-dividing state, okay? G2 is going to take place after S phase. So if you remember, so the G1 checkpoint is in G1, and then we go through S, and then we go into G2. So during G2, we're going to make sure that all the DNA has been replicated. Did I make a copy of all the DNA that I now have two full sets of the chromosomes, okay? And is the DNA damaged? Is there anything majorly wrong with the DNA so that there would be a problem if my cell divided, okay? If it detects problems with the DNA, oftentimes it's going to halt it. There's this, I can't think right now what the word is, but it's P53. It's going to be a gene that's going to basically stop the cell cycle so that you don't replicate until it's able to fix that DNA. Oh, here it is right here, P53. And so if it detects that damage, P53 is going to come in. It's going to make something called a CDK inhibitor. And the CDK inhibitor is then going to bind to the CDK. CDK is cyclin-dependent kinase. So again, we're looking at a kinase that phosphorylates. And that's what you see in all of these, that you're going to have some type of CDK. They're not going to ask you the specific CDK, but they may ask you about CDK. And so what's going to happen is that it's going to halt the process until you have a second to fix the DNA and make it so that you can replicate. Okay. There's a question: does the cell enter G0 and G2 if the DNA is damaged? No. If the DNA is damaged, it's probably going to try to repair it. So we haven't gotten to unit 6 yet, but there's something called DNA polymerase. DNA polymerase is going to be the thing that makes the copy of the DNA or makes that DNA polymer. And so there are multiple types of DNA polymerase, and one of those is able to go in and fix the problem. So sometimes we'll use endonucleases. Those are going to be enzymes that will go in and cut. And so it'll cut out that segment of DNA that's wrong, and then it will then replace that segment if it needs to be replaced. So there's a lot of different ways, cellularly, that it can fix the problem. You're not needing to know all of that information. You just need to know that, hey, if everything's wrong in G2, it's going to hold it until it fixes that. So someone says, what's CDK? CDK is cyclin dependent kinase. And so what it is, it's a protein that's going to kind of, it stays at a constant level. And as you go through G1, S, and G2, the cyclin level is going to increase until we get to a certain point. And then the cyclin-dependent kinase will start to work. It's an enzyme that will phosphorylate, that will allow you to move through different processes. Okay. And then when we get into our M checkpoint, the M checkpoint is making sure that every single sister chromatid is attached to a spindle microtubule. Okay. As we said before, you've got two sets. You've got the original chromosome, right? And then you have the duplicated chromosome. These are sister chromatids, okay? So if we move, and I'm going to change my hand to this way because you can use my arm as a microtubule, okay? So if we were to think for a second, like here's our microtubule, right? And I've got two of them, and for some reason, one of them isn't attached, right? So if you go through the process of cell division, right, and you try to divide the cell, both of these chromosomes are going to move to one side. So the cell over here is going to end up with an extra chromatid, right? An extra amount of that certain chromosome, right? And over here, we won't have that chromosome at all, which would be a problem. So the M checkpoint is making sure that there is every single microtubule is attached to every single one of our sister chromatids so that when we go through the splitting process, we can evenly split those chromosomes to opposite poles, okay? And so they're going to work through a whole different processes. You don't really need to know about the cohesin nor the separase, but that's what's happening is there's these cohesin molecules that are holding them together that then causes the release of a certain enzyme, which then releases the separase that then allows you to split the two, okay? So there's that. If there's an error that's unable to fix, it will perform. Yeah, it'll go through apoptosis if it can't fix itself. It's too late to go through zero. Yeah, it's too late to go through G0, so it'll actually just bye-bye. So that brings us to the end of the content, okay? So I have, I think, 3 multiple choice questions and then 1 free response. We'll do the 1 multiple choice, then we'll do the free response, then we'll come back and do 2 multiple choice. So I'm so sorry. So what I like to try to do is show you different like ways to work through problems. Okay, so here in this one, we're going to kind of analyze the answer choices to try to figure it out. The M checkpoint is actually during metaphase. It's like after metaphase before you go into anaphase. And so we're making sure that everything has been attached. So it's going to take place in metaphase. So it's a little easier to remember. The M checkpoint is after metaphase, like M and M. I don't know. Try. Anyway, so insulin's protein hormone is secreted in response to elevated glucose levels. So if our glucose level is too high, insulin is released. Cool. When insulin binds to its receptor on the liver cells, the activated receptors stimulate phosphorylation cascades that cause the translocation of glucose transporters to the plasma membrane. Okay, so what this tells me, okay, so when ligand binds to a receptor, well, the thing that we know that binds to the receptor is going to be your signaling molecule or your ligand. So based on the information it's given me so far, insulin is a ligand and it binds to a receptor. After it binds to that receptor, I'm going to see the transduction, which is a phosphorylation cascade, and then the response is going to be translocation of glucose transporters to the plasma membrane. Okay, so we're analyzing this, we're bringing this all together. Okay, so based on the information provided, which of the following best describes the role of insulin in this liver cell signaling pathway? Now, without even looking at the answer choices, you all can tell me what insulin is. Now, if you want to go ahead in the chat, you can throw in what you think insulin is based on the fact that we've just gone over it. But let's look through our answer choices just in case you are clueless. So it says it acts as a ligand. Well, what is a ligand? Well, a ligand is a signaling molecule. So the ligand is the thing that's going to be binding to the receptor that's then going to cause a response. Well, it says right here, insulin binds to its receptors. I think A is the answer, but let's just double check. It acts as a receptor. No, it tells me right here that it's binding to a receptor. So it can't possibly be the receptor if it's binding to a receptor. So a receptor is a protein that the ligand is binding to. It can either be cell surface or it can be intracellular. Okay. And so based on that, I do not think B is the answer. We're going to cross B out. Okay. It acts as a secondary messenger. Well, secondary messengers are part of transduction. And this clearly happens before transduction, plus the transduction is a phosphorylation cascade. So I don't think that it's going to be transduction, right? I don't think that it's going to be a secondary messenger. And then D says it acts as protein kinase. Well, I saw protein kinases in the phosphorylation cascade. That's how we got the phosphorylation cascade. But again, the insulin is before we even get to this, right? Because it said the receptors stimulate the phosphorylation cascade. So the insulin isn't part of that process. And so I don't think that it's D either. So that tells me that the answer, of course, would be A. Now, as I said, y'all could have blocked all these answers and you could have come up with this on your own without even like reading the answer choices. And so that's a good strategy, because if you can come up with the answer on your own, a lot of times that's the right answer because you didn't, like, the test didn't confuse you, make you think something else. Sometimes when you read the answer choices, they all sound right. And you're like, A, B, and you get confused. So if you can come up with the answer before looking at the answer choices, that's a good way to, like, oh, here's A, let me move on with my life. Make sure you do read all the answer choices just in case, though. So somebody says, do we need to know about tyrosine kinase proteins and how they come together to form a dimer? Tyrosine kinase is not in the CED. It is a example of a receptor. So my assumption is that if you have a tyrosine kinase protein on your exam, they're going to have to explain to you that it's a receptor, or they're going to have to show you a diagram that shows what is happening. So somebody says, where's practice problems coming from? I'm pulling them from my review guide, but my review guide was built using all of the released multiple choice and free response questions that they have out there. So College Board has questions in their CED. They have questions in the 2013 practice exam that's openly accessible, as well as there's a new CED. Like, so every like available question that I have that aligns to the standards I've put into there. I also told you where it all came from in the like answer key. Sorry, y'all. Okay, so usually explain it as FRQ. Yeah. So do you need to know about telomere and telomerase? So telomeres are usually going to be more looking at meiosis. I don't believe they're in the CED, but the telomere is a section at the very end of the chromosome that would get smaller every time you go through DNA replication. And so telomerase's job is just to replicate, make that longer in your gametes. But we really don't need to know that in Unit 4. Yeah, okay. So we did all that, moving on. Okay, so here is the 2022 question number 1. I really like this question because it's going to bring in some stuff we learned in Unit, I think, 2, Unit 1, and it's also a really great example showing signaling transduction pathway. Okay, so here they've given us this diagram. And they've talked about this pathway right here. It tells us that there's an extracellular ligand. Here you see it. It binds to the G protein coupled receptor in the plasma membrane of the cell, which then triggers intracellular signaling, as you see in figure 1. Okay, A. After the ligand binds, the GTP is replacing the GDP that's bound to this GSA. You see the GSA right there. A subunit of the G protein. This causes GSA to activate other cellular proteins, including your adenylyl cyclase that you see right here. That then activates your protein kinase. In the cells that line the small intestines, this pathway is going to release the chlorine ions from the cell under normal conditions that hydrolyzes the GTP to the GDP, thus inactivating the adenylyl cyclase, which then stops the signal. Okay, so to give you a lot of information, all of this verbal corresponds to this diagram, and you can kind of see where each of those components are. Okay. So then go on and talk about individuals that are infected with a certain bacteria experience severe loss of water from the body, which is dehydration. This is due to the effects of the cholera toxin in these intestinal cells. The scientists then studied the effects of the cholera on 4 samples of isolated intestinal cell membranes containing the G protein related signaling transduction pathway shown in figure 1. So GTP was added to samples 2 and 4. So you can see this little this positive here showing us that GTP was added. And the cholera toxin was added to samples 3 and 4. So again, you can see the plus right here showing us that they added cholera toxin. The scientists then measured the amount of cyclic AMP produced by the adenylyl cyclase in each of our samples. Okay, so just looking at this alone, I can already tell a lot of information. I can tell that the independent variable, what the scientist is changing, is going to be the presence or the absence of the GTP, the presence or the absence of the cholera. I can also tell about what the dependent variable is. The dependent variable is going to be the rate of the cyclic AMP production. That's what the scientist is going to be measuring. Okay. And FRQ would recommend reading the description first of the diagram to save time. I usually go and look at what the question is asking. In the new redesign, they've already broken it up. So like the first question usually doesn't have anything to do with the actual like prompt. It's just going to be a content question. And then B is usually specifically asking about it. And so they've broken it up so you'll just have what you need directly above it. So it'll make it a little bit easier to work through all that reading. Your teacher gave you this exact question. Yeah. Okay, so moving on. So the first part, as I said, this is going to be a content question. It is just asking you about biology. And part A is always going to just be a straight biology question. And so if you read this and you're completely confused and you don't know the answer, you are welcome to skip part A. Just skip A, go straight to B, right? So yeah, you can have more than one independent variable. So describe one characteristic of a membrane that requires a channel to be present for chloride ions to passively cross the membrane. So thinking to myself, okay, so chloride ions, an ion, and as I go through these, you're welcome to answer in the chat yourself. Usually by the time that I get to the answer, you've already had a moment to see the question and then also answer it yourself. So you're welcome to type in the chat and try to give an answer. So chloride ions, I know that ions are going to be charged items. I don't really need to know if it's positive or negative. I just know that those are charged. And if there's a charge, that means that it's going to be polar. Now, so what this is asking me is a characteristic of a membrane that requires a channel to get something polar across the membrane. Well, polar materials aren't going to go through something that is non-polar. So what is the characteristic of the membrane? Well, the membrane is going to have to be non-polar, right? So if you look here at our membrane, you can see we've got these little channel proteins. And so it's saying, why do I need this channel protein? Well, because this area right here, all of this little part, this the fatty acid tails of your phospholipid, these are nonpolar. And because of the fact that membrane is nonpolar, this charged material is going to have a channel to get through that membrane. And so you can talk about how the membrane is nonpolar. You can talk about how the membrane is not charged. You can talk about how the membrane is hydrophobic. And so the interior of the membrane or the phospholipid tail is nonpolar. The interior of the membrane or the phospholipid tail is not charged. The interior of the membrane or the phospholipid tail is hydrophobic. And as you go through these, remembering that you must have whatever is underlined. And so if you're talking about something being nonpolar, you can't just say it is nonpolar. What is it? I don't know what it is, okay? So you need to be very specific, and it was specifically talking about how the interior of that membrane is nonpolar and hydrophobic, and because of that, chlorine ion that's negatively charged can't get through. So good job, ions are polar, so you need the channel protein. This is the nonpolar interior environment of the, yes, okay, so nonpolar interior layer, since plasma membrane is amphipathic. So talking about the amphipathic part might be a little sketchy there, okay? I'm glad you know that word, but we need to make sure we are talking about the interior of it. So good job on talking about the nonpolar interior layer, okay? Nonpolar lipid is going to repel the ions. Yeah, I guess so, because you're talking about the nonpolar tails, so that might work, yeah. Okay, so the second part asks us to explain why the movement of chloride ions out of the intestinal cells leads to water loss. Okay, so they're saying, okay, we've told you that when chlorine leaves, that water's also going to leave. So you're not going to tell me because it loses water, because it's telling us in the question that it loses water. So explain why chloride leaving causes water to leave. Okay, so if I have a solution, okay, I have a solution. And inside the solution, there's a whole bunch of ions, right? And then outside, there's not those ions, right? We've got, right now it's isotonic. We've got the same amount on the inside, same amount on the outside. We're good, right? So if some of these charged ions leave my cell, right? They come out into the extracellular area. They're now out on the outside, okay? The inside of my cell is now going to be hypotonic to the outside, right? Because the chloride left, making the outside hypertonic and the inside hypotonic. So, of course, water moves from areas that are hypotonic to hypertonic. And so we're going to see that the water is going to move out because the space outside the cell becomes hypertonic or hyperosmotic, if you've learned that word, compared to the cell. And so the water moves out of the cell. The space outside the cell is going to have a lower water potential compared to the cells, so the water moves out of the cell. Okay, so keep in mind that the water potential has to do with the potential for water to move. Inside the cell has a high water potential. Outside the cell has a low water potential. So water moves out of the cell from the high water potential to the low water potential. Outside the cell becomes hypertonic. Great job, great job. Okay. So now it tells us I need to identify an independent variable in the experiment. So first think, what is an independent variable? Well, the independent variable is what variable was modified by your researcher or scientist. This is going to be given to you in the prompt. Somebody says, what's the difference between hypotonic and hypertonic? We did that in unit 2. Hypotonic has to do with there's a low amount of solute and a high amount of free water. And so water is going to surround each of your solute molecules, each of your solute particles. And so the free water is the water that's available to move. And so with hypo, we think about being low amount of solute, so there's a high amount of free water, and then hypertonic kind of makes us think that there's a whole bunch of solutes in it, so there's less free water, so the water moves from the hypotonic to the hypertonic. Okay, so independent variable, oftentimes you can get this just looking at the prompt. So if you have absolutely no idea what is happening, you can look back at the prompt and say, okay, what did they change? I can look at the graph. I can see or table. What did they change? What was modified by the scientists that allowed me to have my experiment take place? Okay. So if I look here, I can see, okay, well, I have these 4 different samples. That's not really anything. That's just identifying them. GTP. These two have it and these two don't have it. Okay, cool. So maybe the presence or the absence of the GTP. And then here, same thing, the presence or the absence of the cholera. Okay, now you had to mention presence or absence. Okay, you couldn't just say, was there cholera? Because that's not really telling me anything, okay? So you could either say the presence or absence of GTP and cholera, or you could have put those as their own separate thing of presence or absence of cholera, presence or absence of GTP, okay? But you had to be very specific of presence or absence of those, okay? You couldn't just say the absence of it or just saying the presence of it, okay? So great job to everybody in the chat. I see a lot of y'all talking about the cholera and the GTP. Yes, this was the AP exam from 2022. Okay, so this was a release question. The exam for 2026 is new. It's a new CED, but I believe this is still applying because this still is within the same ideas. Okay, so our second part of this question says identify negative control in the experiment. Negative control. Okay, what's the negative control? Negative control is going to be where it doesn't get whatever the experimental treatment is, okay? So it's not treated with the actual independent variable, okay? So somebody says, does inclusion or exclusion of cholera toxin count, or does it need to be presence or absence? Inclusion, exclusion might talk about presence or absence, or the, adding it or not. I believe that probably would be acceptable on the exam. So I wouldn't really, I think that would be good. But since they gave us in the chart presence and absence, I would probably use the terms that they gave me, just because if you use the same words they use, oftentimes it's a little bit more aligned. So it says identify negative control. So if I look here, I can say, okay, well, what does not have my treatment group? Well, my treatment group was the cholera and the GTP, and I was seeing what would happen when those two things were added. So sample 1 would be a great example of my negative control. But here's the thing. Sample 1 is missing both of those. But what if you as the student didn't say, oh, well, both of these are independent? What if you just thought one of them was independent? Well, if you thought that the presence or absence of cholera was your independent variable, then you could have said, 1 and 2, because the fact that those ones are going to be the ones that don't have the cholera. Or if you were talking about the GTP, presence or absence, being your independent variable, then 1 and 3 could be your negative control. So there's a lot of options you could have put here. You could have been sample 1. You could do 1 and 2. You could do 2 alone. You could do 1 and 3. You could not do 3 alone because of the fact that the next question is about to tell you that sample 3 is a control. So you could not do 3 alone. But you could talk about sample 1 and 2, samples 2, and then 1 and 3. And you were able to describe the sample as having, lacking both components, or lacks just the cholera, or lacks the cholera and contains the GTP, or lacks the GTP. So that kind of encompassed all of those options. So that you really had a lot of options you could choose here. So then the next part of this question says, justify why the scientists included sample 3 as a control treatment. And so I'm looking at 3. I'm saying, okay, well, why would 3 be considered a control? Like, what does 3 have, or what can I compare 3 with? So 3 doesn't have the GTP, but it does have the cholera. And I can't really, if I compare it to this one, that will just tell me what happens with cholera. So I really wouldn't talk about that one alone, because this one doesn't have either GTP or cholera. But what could I compare it with? Oh, I can compare it with 4. So I compare with 4, I can see what the presence of GTP does. Do I need GTP for this process? And if you think back to that pathway, you know there's GTP was part of the signaling transduction. So this is going to confirm, do I need to have that GTP for the process? So I can compare the cyclic AMP production with that of the sample that has cholera and GTP. I can compare it to sample 4. Or comparing samples 3 and 4 enables us to evaluate whether the activity requires GTP or not. Sample 3 is a control, yes, but so we need to say why would they use 3 as a control, and so they're wanting you to justify it. We know it's a control. We want to say, well, why am I using this one? And so the reason why is because you're going to compare it to sample 4 to be able to determine the effect of GTP, because if both 3 and 4 both have that cholera. So that's what we see there. So the next question, based on the data, describe the effect of cholera on the synthesis of cyclic AMP. So if I look at my graph, I can say, okay, let's see. Here's sample 1, and then I can compare that with, let's compare it with 3. So if I compare it with 3, here I can see that 1 and 3 both don't have GTP. So it allowed me to compare easily. And then here cholera, and I'm sorry, no cholera, and then cholera. So I can see what is the effect of cholera on the presence of cyclic AMP. So I can see that sample 1 has 0.5 and sample 3 has 0.5. So in the absence of GTP, I can see that there is no effect on the production of cyclic AMP. So those are going to be the exact same if there is no GTP. But wait, there's more. I know you probably don't get that joke, but it's okay. So here if I look at 2 and I look at 4, they both have GTP, no cholera and cholera, so I can see what is the effect of cholera. And look, if GTP is present, there is an effect. There's a positive effect. And so you did have to bring in that GTP component. You had to say, in the presence of GTP, there is a positive effect. In the absence of GTP, there is no effect, okay? You had to bring in cholera toxin, increase the production of cyclic AMP in the presence of GTP. Cholera toxin has no effect on the production of cyclic AMP in the absence. Now, you did not have to have both of these answers, but you did have to have one. And so this was making sure, did you understand which 2 of all this data, which 2 are you going to look at to determine, does cholera have an effect? Because if you didn't appropriately state that GTP, then they didn't know what you're looking at, and then you were not being specific enough with your answer, okay? So I see a lot of no effect or has no effect. Now remember, we do need to have that GTP in there. Cholera has no effect on synthesis. Again, you need to have GTP in there, okay? So now the second part. Calculate the percent change in the rate of cyclic AMP production due to the presence of cholera toxin in sample 4 compared to sample 2. Now here's the thing. You get a formula sheet on the AP exam. It's a great formula sheet. And based on last year's drama, now you will have a printed copy of that formula sheet. Great, awesome. This equation's not on it. So this is just a normal experimental calculation that you would be doing. So you should know this calculation and they shouldn't have to give it to you. So they don't give it to you, okay? So you need to, right now, I need you to memorize this equation. You need to know this equation, okay? The equation is going to be, oh, I'm ahead of myself, sorry. The equation is going to be final minus initial over initial. I'm seeing how much did it change. So I'm going to see how much changed compared to how much it was to begin with. So final minus initial over initial. And then since it's a percent, we're going to multiply by 100. I don't have that on there. I'm so sorry. Anyway, so they want us to compare samples 4 and sample 2. Okay, so I'm going to say, okay, well, before cholera would be my initial, and after cholera would be my final, because I'm seeing what is the effect of that cholera, right? So what I'm going to do, 127, because that's how much I had at sample 4, minus 10, because that's how much I had at sample 2, because that was my initial, divided by how much it was at 10. So I'm seeing what is the percent change when I'm looking at the presence of that cholera toxin. So 127 minus 10 would, of course, be 117 divided by 10 gives you 11.7. And then again, multiply by 100 to get me into percent. So it gives you 1,170%. Y'all, a lot of you freak out when you see a percent that big. And you question, you're like, is that right? Can the number be that big? So sometimes your numbers might be a little big, okay? Don't freak out. Double check your math. Say, okay, did I do the right math? If you did the right math, don't freak out and keep on moving, okay? So this answer should have been 1,170%. There was no other answers accepted. If you use the wrong numbers, if you made a math error, Doesn't matter, okay? This is the number that they were looking for on the exam to get that 1 point. And again, this formula, you must know to memory. Do not forget this formula. Final minus initial over initial. So they then say, okay, well, a drug is designed to bind the cholera toxin before it crosses the membrane. So there's a drug that binds the cholera, so cholera doesn't enter my cell. Scientists mix the drug with cholera and then add the mixture and GTP to a sample of intestinal cell membranes. Predict the rate of cyclic A and B production and it gives us units if the drug binds to all of the toxin. So they want you to predict. So with this prediction, you got to come up with a number, okay? You can't say it's increasing. You can't say it's decreasing because they've given you data, okay? If you look in this chart, can you find the place where you have GTP and you don't have cholera? Because again, it's telling us that the drug is going to bind to the cholera. So if the drug binds to the cholera, there is no cholera present. So you got to figure out, where is it? Could you divide by 10 and then multiply by 100? No, because of the fact that this is 117. So you can't take 117 divided by 10. Anyways. Anyway, so can you find where it is that we've got GTP and we do not have cholera? Well, that's sample 2. Sample 2 is where we have GTP present and I don't have cholera because the drug bound to the cholera, which means my answer is 10. Did you have to do math? Nope. You did not have to do math. You just had to look at it. Don't say around 10. It is 10. The answer is 10. There was no other answer accepted. Okay. And since they gave you the unit in the question, you did not have to put units, but I highly recommend that whenever you're writing an answer, to go ahead and put the number. Okay. 127 you meant, sorry. You can't do 127 divided by 10 because you didn't subtract by your initial. So you're going to have to do the full amount. Okay, so the last part of this question. In a separate experiment, scientists engineered a mutant adenylyl cyclase that cannot be activated by the G whatever. Okay, the scientists claim that cholera toxin will not cause excessive water loss from the whole intestinal cells that contain the mutant adenylyl cyclase. They wanted you to justify. Now here's the thing. They're telling you what's going to happen. They're telling you that it will not have water loss. They told you there's no water loss. Why is there no water loss? Look at the pathway and tell me, why is there no water loss? Okay. Now it takes a second and you have to walk through every single step of this pathway, right? So if I have a mutant adenylyl cyclase, okay, then adenylyl cyclase is not there. So there is no adenylyl cyclase. If there is no adenylyl cyclase, then you're not going to be converting cyclic AMP you're not going to be converting ATP into cyclic AMP. So cyclic AMP also doesn't exist, okay? So if you have no cyclic AMP, then you're not going to activate protein kinase. And if you don't activate protein kinase, you don't release chlorine. And if you don't release chlorine ions, you don't lose water. Okay, that is what you had to walk through, the entire pathway. Cyclic AMP will not be produced, the protein kinase will not be activated, and the chloride ions will not be secreted. So less water will then leave the intestinal cells. Okay, so this was justifying their claim. They gave you a claim, use the evidence to then support that claim and explain to them how that claim is true. Okay, so next question. Platelets are fragments of larger cells and normally circulate in the blood without adhering to blood vessel walls. Ms. Jones, I thought animal systems wasn't on the exam. Animal systems is not on the exam. But what happens is they can give you examples and then you have to apply the biology to those examples. So do not freak out if you see questions that aren't things your teacher has taught. Do not come back and say, your teacher didn't teach you things. They did. They taught you a lot of things, okay? You're needing to apply your knowledge to new examples, okay? They don't want you going in already knowing all the answers. They want you to apply what you know, the biological concepts, to new ideas and new problems, okay? So when the wall of the blood vessel is damaged, collagen fibers in the wall are exposed to the interior of the blood vessel. The exposed fibers and chemicals released from the endothelial cells and line the blood vessel attract platelets, which then start to form a plug and release other chemicals. Okay. So based on this diagram, you can see this whole bunch of things they just talked about. You can see here that there are these platelets. You could also see that there's all these like released chemicals. Little dots are released chemicals. And so from that, we're seeing that we're going to form this little like plug in our blood vessels. Okay. So let's see what's going to happen. Okay. So which of the following best explains the feedback mechanism illustrated in figure 1? Okay. So they want to know what kind of feedback do I have going on? So this is an example of positive feedback. Positive feedback. Because a few platelets that initially bind attract more platelets to the damaged area. So positive feedback is going to amplify. So if I have a couple platelets that were in the area, and those couple platelets release these chemicals that then cause more platelets to come to the area, that would be positive feedback. Yeah, I like A. A sounds good. Okay, let's keep looking. This is an example of positive feedback. Okay, I've already agreed that's positive. Because it results from the interaction among collagen, endothelial cells, and platelets. The collagen and endothelial cells don't look like they're doing anything. They're just like chilling and they've just been broken. So I don't really think B is an answer. B is not very good. This is an example of negative feedback because a large clump of platelets can block the blood vessel and prevent blood from flowing through it. Yeah, like we do have a clump of those, but there's no feedback taking place because the platelets are like communicating to form that plug. And the feedback that we're seeing is the making of that plug. So I don't really like C either. D, this is an example of negative feedback because the accumulation of platelets returns the open blood vessel to the closed state. You're right, we are seeing that we're closing it back up, but that's not really what's allowing it. Like, how did I get to the accumulation of platelets? So our appropriate answer here is A, and a lot of y'all said that in the chat, so great job, y'all. And now our last question. And I really didn't have a lot of questions on mitosis, so this was the best I can come up with. I'm sorry, y'all. If chemical signals in the cytoplasm control the progression of a cell to the M phase of the cell cycle, then the fusion of a cell in G1 with a cell in early M phase would most likely result in the. Okay, so the M phase was when we were going through mitosis, right? The cell was doing prophase, metaphase, interphase, telophase, right? Okay. And G1 was during interphase, like before we even had anything taking place, okay? So we're saying there's all these chemicals inside that's allowing for the communication. So if I have a G1 cell with an M cell, what's going to happen? Replication of chromosomes only in the G1 cell. Well, if they're together, I'm not going to have replication of chromosomes happening in one or the other because of the fact that they're now together and they're using the signals from one of them, right? Exiting both cells from the cell cycle and going into G0. Now, M phase 1 is already going through its process. Condensation of chromatin in preparation for nuclear division. Well, I do see that this one's an early M, right? So we're going to need to like condense to go through go through division. So C sounds pretty good. Transfer of organelles from G1 to the M phase. We're not going to see that those are just going to jump ship. So the only logical answer we have, and I see all y'all are putting little question marks there, but of course be C. C is our answer here because that's the only one that's talking about like what's happening in the M phase and what's taking place and how we would get to that process. So replication of chromosomes only in G1 is not logical because they're now together. B, you wouldn't really exit it. We don't have the signal that's going to cause it to exit. We're already past the G1 in this one. So we're already going through the process. So we already have the internal signal to say, hey, keep going. And then transferring of the organelles from one cell to the other isn't really what's happening because like we're already past that. So that was all of our example problems. In terms of the exam, some of you have already heard this, but of course, y'all ask new questions sometimes. So I'd say this every time just because I want to make sure that everybody knows what the format of the exam is in case this is your first time turning into one of my videos. So the exam is hybrid, okay? It is a hybrid exam. You have both physical and you have digital components, okay? So Blue Book is what you're going to use. So you should have Blue Book already downloaded on your device. If you don't already have Blue Book, go ahead and download it. There are practice questions in there so you can get a feel for it, okay? You definitely need to make sure that you know your username, okay? Know your username and password. When you go to that test site, okay, they're going to make you lock everything up. Your smartwatch, your phone, everything is going to get locked up except for your device you're taking this exam on, okay? So you need to have that username and password memorized, and you need to know how do you log into AP Classroom, okay? A lot of y'all have your stuff saved. You're not going to be able to access that when you go into the Blue Book app, okay? So you definitely need to know this information, okay? I'll come back to the questions at the end, I promise. So in terms, and when I mean questions, I mean like questions that aren't about what I'm currently talking about. So section 1, the entire thing is going to be on Blue Book, okay? You will have questions in there, multiple choice questions. There are going to be 60 multiple choice questions. You have 90 minutes. The timer is embedded. You'll see it at the very top of your screen. you'll answer all those questions in there. So everything from Unit 1 is there, okay? In Section 2, only your questions are in Blue Book and your timer is in Blue Book. Your written responses will be in a book. And I have an example of what that book looks like in a second, just so we can talk about it, okay? In Section 2, you'll have 2 longs, 4 short FRQs. You have 90 minutes, okay? My advice to you is to take the first 5-ish minutes and look through all the questions. Renumber them. Say, hey, I know this long FRQ, I know these 3 short FRQs, I know this long, I can renumber them, decide what order are you going to go through those questions. You have scratch paper. Write on your scratch paper, which are your order. You can number them on your paper. I really don't care how you number it, okay? But come up with what is the order you're going to do these, okay? So give yourself about 5 minutes for that. You want to spend about 20-ish minutes on your long FRQs, and you want to spend about 6 to 8 minutes on your short FRQs. That will give you enough time to be able to go back to previous questions if you need to go back to any of them. It also allows you to answer something you know about every question. Okay. It also allows you to be able to reread. Sometimes when you reread your responses, you're like, wait a minute, that wasn't right. And you can correct it before it leaves you, right? So that's my advice. You do you, I really don't care. Okay. In terms of G2, okay, I'm sorry, question 2. Question 2 is going to be your graphing question. You will have to make a graph. Please, 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 please use pencil. Don't use your pen on the graph. Use a pencil. And by using a pencil, that allows you to make a mistake and erase it. Okay. You can cleanly erase it with a pencil. Okay. Everything else, let's make it a pen. But in terms of your graph, please make that pencil, okay? Your school doesn't use your devices, so I don't know what you're going to use, but you can still download it on your own device to practice. And yes, your College Board login is the same for your Blue Book, okay? So the exam is in 50 days. Yes, 50 days from today is when your exam is. May the 4th be with us. In terms of line graph versus bar graph is going to be categorical data, and line graph is going to be like longitudinal data, like you can visibly see the difference. I've got a couple different examples. You're allowed to use pencil for the entire FRQ section. That change was made last year. You can use a pen or a pencil. I recommend using pencil only on the graph, but you could technically use it for all of them. So, but I'm saying that if you're in the middle of writing an FRQ and you look down like, oh, I have a pencil in my hand, oh no, do not rewrite. Just pick up the pen and just keep on rolling, okay? Do not freak out. Okay, so there's that. Are you allowed to bring a pencil? Yes. Can you tell me? Yeah, what can I do? Okay, so where did I put the thingy? Oh, I must have put it out of order. Let me look at this. Okay, so this is what your booklet is going to look like. Okay, at the top is going to say, begin question 1. Begin your response, question 1, let's just And it says, do not skip lines. Hey, you see this big do not skip lines? That's talking about in between every single thing. If you write a paragraph and then you skip a line, and then you write another paragraph and then you skip a line, don't freak out. Please just, it's fine. Do not rewrite. None of that. Okay. You're perfectly fine. Okay. Then here's question 2. You're going to have a graph. It says on here to use only one color. The scans are black and white. Your readers cannot see to use the blue pen here and a black pen there and a pencil here. They can't see the difference. It is literally just a scan. Okay. So make sure that you have a key for it. Like, so you've like shaded one and not shaded another. You've dotted a line. You made little hearts if you wanted to. I don't really care how you separate the two. But depending on your graph, you're going to need to have some type of key or some type of labeling if you have different things that you're going to need to identify based on, I guess you would do color sometimes, okay? And then you, of course, have question 3. Now, here's the thing. Students sometimes will flip the page, and they've already written question 2. They've already answered question 2. They've already done all this stuff, and then they realize, oh, my God, I'm on the wrong page. Do me a favor. Please do not rewrite your response. Go to question 3. Go to question 2. Go to wherever question you were currently supposed to be on, okay? And say, see question wherever you wrote it. And the reader will be able to find your response. Please do not rewrite your response. I'm repeating myself, and I'm trying to get this word out there to the public, to the world, as soon as possible, okay? Do not, I repeat, do not rewrite your response, okay? Go to wherever it's going to be and say, if you wrote question 3 and question 2, please see question 2 for response. And your reader will press the button that says full book, and they will go to find question 2 and they will read it. Now, they're supposed to go full book if you don't have the right answer there anyways, okay? That's what they're supposed to do. but it will make their job easier and it will ensure that they know where to find that question. Okay. You have to keep in mind, they're scrolling through them all, searching for where that response is. They could accidentally miss it, right? We are all human. So if you tell them where to go, there's a higher chance they'll be able to find your response and it will save you from having to rewrite. Okay. So let's go back here. So helpful resources. What resources can you use? Okay. Number one, hi, me, AP Bio Penguins. I know, duh. Anyway, so there's a ton of resources on my website. Every single thing is free. I do not charge for any of my resources. I want you to have whatever you need within your hand to be successful on this exam. Okay. AP Classroom. There are these performance progress checks your teacher can assign to you. If your teacher hasn't at this point, say, hey, can you please open those up? I'd like to use them to practice. The best multiple choice questions you can use to practice are the ones from College Board. They were written from the same people. They've gone through the same level of scrutiny. They've gone through everything that the College Board questions have gone through with the same hands. So I highly recommend that you get a copy of those PPCs, okay? Now, those of you that are self-studying, somebody somewhere, there's a test coordinator, there's someone who has you in their AP Classroom. Figure out who they are and ask them to just open them all up. At this point, we're 50 days away from the exam. You're not screwing up anything in your classroom. Just say, hey, ma'am, hey, sir, can you please open them up? They don't have to be a grade. Well, actually, they can be a grade. But can you please open these up? I'd like to practice, okay? Also, your teacher has access to practice exams. There are 3 2020 practice exams that they have access to. They can proctor them. For me, I'm going on April 18th, and I'm going to give my students a practice exam. They're going to sit down at 8 a.m., and they're going to have to sit through a 3-hour exam so they can feel what that mental anguish feels like. And they can go through the process of working through the online submission of the test, as well as the handwritten, and there's a whole back and forth that they're going to have to go through, okay? Podcast. If you're like, hey, I just need to listen to someone talk, like I need something. The Absolute Recap. The Absolute Recap is, my good friend's Melanie King. She, again, everything is free. I'm all about free over here. So she's got worksheets that align. So if you're like, I need to, write something down, I need some worksheets. She does have worksheets. But if you're just like, I want to listen, I'm on my way on the bus, I need to listen to some bio, I'm, walking my dog, I'm whatever, like I want to listen. Then you can do the absolute recap. In terms of review books, my favorite is the Barron's book, okay, by Mary Worth. That's my favorite. But you know what? Everyone has their own opinion. Everybody has their own favorite, okay? Go to the bookstore, pick up a Barron's book, pick up a Princeton book, pick up a 5 Steps to a 5, pick up whichever book you want, okay? And read through a section and see which book explains things the best to you. The questions are all approximately on the same level in terms of those review books. They all approximately have the same number of practice exams. They're basically the same. It's just the author. Which one is going to speak to you better? Which one is going to make it so that you better understand? That is the point of a review book, is to better understand the content, okay? So pick one that actually works for you. Again, I already did that. So now in terms of the exam, I've already got, the Wayground game. So you can scan that little QR code. It will bring you, I think, to the page that has all 120 of my Wayground games. Okay. And of course, I'm doing my daily review. Okay. And on my website, is where all my resources are. Okay. So I will be back live on March 22nd. Yes, next weekend. There is NOSB, National Ocean Science Bowl, next weekend. I don't know whether we have made it to the round robin. I don't know if I will be home by 3 o'clock. My goal is to do the session at 3 o'clock. I am human. I have to do 20 million things at once. And so there's a chance that I won't. And just like today, when I wasn't able to make it here on time, I did post a little comment on there. I posted it all over my Instagram and I put it on TikTok. I put it everywhere that I wasn't going to make it in time. So look for a message from me if I'm going to be late. If anything, the session is recorded. I know some of you have probably already left me by now because I've already gone over my hour. But it's there if you want it. So hopefully I'll be live. Yeah. Okay. This was my 8-week session. There's tons of review things on there, so you can use that. Okay. So somebody says, okay, here's the question. So what is Blue Book? Blue Book is going to be the application that you use. You use the same application when you did your SAT, your PSAT, and now your AP exams are all on Blue Book. So most of the exams are going to be fully virtual. So you'll do like your history exam is all on the computer, if I remember correctly. And so you're literally just going to use that application to take the exam. Can someone please tell me, is the Barron in solving multiple choice? So doing the questions in the Barron's book is helpful. You're definitely going to get review. My students are required to do every single multiple choice question in the Barron's book. So definitely, I believe that it is a helpful resource. So, easy peasy there. Okay. But Is it enough? Everyone is different. It's hard for me to tell you, hey, yes, if you do these 5 things, you're going to make a 5, because everyone has different requirements and different needs. Okay. So do I have any resources for my stats? Not necessarily. I do have, where I've done every of the FRQs. I've gone through the Math Mondays, in which I explain a lot of the statistics. I've gone through Math Mondays, where I've done a lot of the math for the statistics, but I don't have an official statistics unit, since there isn't an official statistics unit. Just wanted you to let you know that I have AM on there. Where do I have AM? Oh, on the schedule, yeah, I can fix that. I'll fix that. I hope I can fix it. I'll do my best. Anyways, okay, so what other questions do we have? Due to an exam conflict, you have to take the International AP Bio exam. Do these exams follow the same format? You wouldn't take the international exam unless you're overseas on day of. I think you mean the alternative exam, and yes, the alt exam is going to have the same format for the multiple choice and the multiple choice, as well as the international exam. But there's no way you would be taking the international exam if you have a conflict, unless you are overseas, and that's the only reason you would take the international exam. Can I do a video on graphing? I do videos all the time. So if you go to the FRQ Fridays, you'll see all of those videos that are on graphing. If you go to my math minutes from last year, I did some graphing ones there. There's honestly a ton of them. Okay. So are P53 proteins always bad? No, P53 are actually good. You want P53. If there's a mutation in the P53, that's when the problem is. So if P53 is not able to stop the pathway and be able to replace your DNA damage, then you're going to have a problem. So like P53 is good. We just don't want it to get mutated. Okay, so can you find old question papers? Yes, so I have all of the old FRQs and all the multiple choice are in my review guide, and they're broken up by the units. So you can go there and pull all of those. Can you cram? Yeah, you can cram the whole unit. I don't recommend cramming, but I mean, like, you're still far enough away from the exam that you can learn the information. Okay. Are there full practice exams? Yes, your teacher can assign them to you and proctor them to you. Daily review is every day I post questions in my story and just try to help y'all overall with the content. They are just quick questions. They're not actually like AP level. So I hope that this was helpful. And I'm sorry for my delay today, but remember, you are AP Bio Penguins and you are dressed to success. And you are going to rock on May 4th. I am killing time at the end of the video because I know that every other time I press the end button and then my little line didn't happen in there. So,